Hi everyone, and welcome to our second live webinar of Immunoassay Applications Week. So this week we're hosting live workshops focusing on different tips and best practices for specific immunoassays. So today's focus is of course immunofluorescence, and I'm really excited to have Dr. Afrida Rahman-Enyart with us today, who'll be leading today's workshop. But just before that, I'd like to do a quick introduction to Proteintech and share some of our latest innovations. So Proteintech is a leading life science reagent manufacturer. We were founded in 2001, and at that time, we were primarily a supplier of antibodies. So since then, we've now developed antibodies against over 14,000 human proteins, which covers about 2/3 of the total human proteome. We also offer immunoassay kits, including ELISA kits, IHC kits, cell separation kits, and antibody labeling kits. But since then, we've expanded our portfolio, and so now we've got many more product lines. We have our humankind branded range of recombinant cytokines and growth factors, and importantly, all of our recombinant proteins are expressed in HEC293 cells, so you can be confident that they have the native folding and post-translational modifications that you would expect to see in human proteins, which ultimately increases their bioactivity compared to proteins raised in E. coli systems. We can manufacture all of our recombinant products at either researchers only or a GMP grade for use in cell and gene line therapies. We also have our Chromotech branded nanobody-based reagents. And nanobodies are really interesting. They're about 10 times smaller than traditional antibodies and combine to their targets with extremely high affinity, making them ideal for immunoprecipitation, super resolution microscopy, and for protein purification. And finally, our genomics line manufactures antibodies conjugated to oligonucleotides, which allows you to perform protein quantification during your RNA sequencing. So all of our award-winning products are self-manufactured. We offer the largest portfolio of antibodies for the human proteome, and we've got a rapidly growing recombinant portfolio. For convenience and testing, most of our antibodies are also offered in 20 microliter trial sizes. We fully understand the importance of high-quality product validation, so whenever we're testing new products, we validate with relevant sample types, appropriate experimental controls, and we validate using knockout and knockdown models wherever possible. And all of that validation data is open access to all of our customers. We're a company of scientists, so we have a good in-depth knowledge of immunoassay applications, and we can offer outstanding technical support for all of our customers. And finally, we have 7 inventory sites and manufacturing and research on 3 continents, so we can offer next-day delivery to many countries across the globe. And I just want to highlight that we define our success as a company through our customers' scientific success, specifically by helping you publish impactful research faster. We currently have over 350,000 citations globally, which makes us one of the top 4 cited antibody companies. And I also just want to highlight that as a company, we strive to make the searchability of our products as user-friendly and informative as possible. And we've launched some really interesting new validation initiatives to help you guys select the right antibodies. So we've experimentally mapped our recombinant antibodies, and we're sharing that information through our unique 3D epitope viewer. So this is a tool that allows you to interact with your epitope structures and explore antibody binding sites. We've also introduced advanced filter options to help you find exactly what you need, such as our immunogen selection function. So this is a selector toggle that allows you to filter for antibodies generated directly from your region of interest. And additionally, you no longer have to filter through all the variations of an antibody from the search results. You can view all formats, including conjugations, buffer formulations on just one page, which ultimately is designed to streamline the process for antibody selection for our customers. And lastly, I just want to highlight that we've built off of the information from our epitope mapper in our database, and we've recently launched an AI-powered search tool on our website called Able AI. So Able allows you to directly chat and ask for product recommendations, help for experimental design, and access relevant validation data, citations, and resources. For example, you could ask Able, please can you recommend me specific antibodies validated for immunofluorescence in your specific sample type, or you could ask Able to suggest optimal fixation and permeabilization protocols for a target protein. And Able will suggest the best product for your experimental needs so that you don't have to spend countless hours searching for the right antibody. Able is a first of its kind tool designed by our scientists in-house with customers in mind, so you can really focus on our discoveries and not on reagent selection. We're very proud to be the first in the industry to launch anything like this, so check it out if you've got some time, either through the QR code on the screen now, or you can find it on our website homepage. And with that, I'd just like to introduce our speaker for today's talk. So we're joined by Afrida, who is the Senior Marketing Manager for Proteintech, to join the team in March 2022. And Afrida is a neurodevelopmental biologist by training, and she completed her dual PhDs in neuroscience and psychology from Indiana University Bloomington. Afrida then did a postdoc at Northwestern University in the Department of Urology, studying the neuromechanisms of pelvic pain, specifically how the gut microbiome and microglial activation modulate symptoms related to interstitial cystitis. Afrida has over 10 years of bench experience in immunoprecipitation, Western blotting, and immunofluorescence, and I'm very excited to hear all about her advice for immunofluorescence today. So with that, I'll stop sharing and hand over to Afrida, but please do pop any questions that you have throughout this talk, either in the chat or in the Q&A box, and we can come on to them at the end. So Afrida, I'll just stop sharing my screen.
Awesome. All right. Well, thank you so much for the introduction, Poppy. And thank you everyone for being here. I'm just going to go ahead and share my screen now. All right. So hopefully you guys can see my screen. So yeah, I'm really excited to be here and talk to you guys about immunofluorescence today and hopefully provide some tips and tricks to help you all with your IF and multiplex IF workflows. So here's just a quick agenda of what we'll be talking about today. So I'll give a brief introduction of immunofluorescence, so a little bit about the concept of how IF is done, and then we'll go through the workflow. After that, we'll go through each of the steps of the workflow, and I'll talk about some considerations that will help optimize each of those steps. And then we'll move on to reagents. So I'll talk about some reagents and new technology that we have here at Proteintech that can help with your IF data. And then lastly, I'll talk a little bit about some common troubleshooting issues and how you can go about tackling these. So immunofluorescence, of course, is a common laboratory technique that allows you to kind of visualize the expression and the localization of proteins in cells or tissues. And so what makes IF so powerful is that it really gives you this snapshot of a very complex biological system. So when you're considering traditional immunofluorescence, you can visualize multiple proteins in a sample by using primary antibodies that are each raised in different species. And so each of these primary antibodies are going to recognize a specific antigen on your protein. You can then use secondary antibodies. And so the secondary antibodies are going to recognize the corresponding IgG of the primary antibodies that you have. And these secondaries are going to be conjugated to fluorophores that can be excited by specific wavelengths of light. And so once these fluorophores are excited by these specific wavelengths of light, they emit light at a slightly longer wavelength. And this is what can be visualized by your fluorescence microscope. So it's really important to note that when you are designing your multiplex IF experiments, that you want to make sure that you're selecting secondaries that have non-overlapping emissions spectra, because your signal might end up leaking between channels and affect data interpretation if you have those fluorophores that are overlapping. So here we're looking at a very general IF staining protocol. So before you even begin your staining process, your first step really is going to be how you're going to prepare your sample. So are you going to use cells versus tissue? How are you going to fix your samples? So if you're using tissue, how are you going to be, preparing it? Are you using fresh frozen? Are you going to be using FFPE? Are you going to use a cryostat or a microtome dissection? So these are all key considerations, and it really is going to depend on what your downstream application will be. So you need to figure that out before you kind of start that IF process. You also might need to do an antigen retrieval step, and we'll talk a little bit more about this in detail kind of in the next section. So after you've prepared your sample, you might then have a permeabilization step. Again, this is really going to depend on how you fix your tissue and whether or not you're looking at an intracellular target. After your permeabilization step, you have your blocking step. So your blocking step is going to be essential in order to prevent any sort of non-specific binding that can occur from your antibodies. So after you've done your blocking step, you're ready for your primary antibody incubation. So this is where your primary will bind to your specific antigen on your protein of interest. And then your secondary antibody incubation will follow. So where your secondary antibody will bind to your primary. And then you'll do any sort of counter stain. So for example, if you want to use Host or DAPI to visualize any of your cell nuclei. And then after that, you have your mounting step. So during your mounting step, you want to use some sort of mounting media or anti-fade in order to prolong your IF signal. And if you're using tissue, generally what you'll do after this is you'll use a cover slip and cover slip your samples. If you're using cells, they might already be on a cover slip, so you just need to kind of put those on the slide and make sure that they're secure. I know commonly people use nail polish to do this, to secure the slides, secure the cover slips onto the slide. And then after the mounting step, you are then ready for your microscopy. So you can go ahead and view your samples and do your microscopy and analysis. So for this next section, I'm going to kind of go through each of those steps of the protocol that I just talked about and talk about some key considerations. So for any IF experiment, it's really going to be crucial first that you know your protein in your sample. So understanding your protein function and its distribution is going to be really important and help you know whether your experimental conditions are accurate. And it's also going to help you optimize your protocol. So it's important to note that, for cell cultures, protein expression and distribution can particularly be influenced by cell health and confluency. So you want to ensure that your cultures are going to be healthy before performing your IF. So for example, bacterial contamination like mycoplasma contamination can actually cause cellular stress. It can result in morphological changes and also changes in protein expression. And this ultimately can then lead to background fluorescence and nonspecific binding. So here in this image, what we're looking at is we're showing staining of phalloidin in HeLa cells. So we're looking at the actin filaments, and you can see here on the left-hand side, a culture that is mycoplasma negative, and you can see that appropriate distribution of what you would kind of expect these actin filaments to look like. And then on the right-hand side is a cell culture that is mycoplasma positive. And you can see now that the distribution of actin looks quite different. And this is essentially going to affect, what your conclusions that are and what you're drawing from your experiments, because you might essentially think that this is the staining that you're supposed to expect, but it's actually not. It's because of the bacterial contamination. The other thing that's going to be important is confluency. So confluency can affect antigen expression and localization as well as signal intensity. So the appropriate confluency is really going to be important for your cell growth, the differentiation, the signaling, and any sort of responses from external stimuli. So here we're looking at an example where we're looking at cells that are 50 to 60% confluent versus cells that are 80 to 90% confluent. And so here we're staining for occludin, and you can see that the ones that are 50 to 60% confluent, the signal is much dimmer, and the expression is not necessarily what you would traditionally expect occludin to look like. So you get that more in that 80 to 90% confluency range. So you just want to ensure that your cell cultures are healthy and happy before you kind of dedicate your time to doing IF. If you're using tissues, you want to make sure that your tissue are not folded or damaged in any way, because these can essentially cause artifacts or some uneven staining. And also, you want to consider, how you're preparing your samples. So for example, if you're planning to use a cryostat and you're freezing your tissue, you need to do a dehydration step with some sort of sucrose solution to prevent any sort of ice crystal formation. And it's also going to help maintain that structural integrity of your tissue as well, because sucrose ultimately is going to maintain that osmotic balance during that freezing process. Another consideration is going to be how you're going to fix your samples. So, fixation is really going to be essential for preserving your tissue structure and your cellular architecture. And depending on your tissue type, it might be recommended to first perfuse the animal prior to fixation by immersion. So, for example, with brain tissue, it's generally recommended that you perfuse the animal. And the reason for this is because you want to remove any of those blood cells because blood cells can interfere with your IF staining and they can lead to quite a few artifacts and background. So that's why, it is generally recommended to perfuse and then further immerse that brain tissue in your fixative. So generally, when you are fixing, there are two main methods that people use. So one is either a formaldehyde fixative, and the other is using some sort of organic solvent. So the way that formaldehyde fixatives work is that it actually creates cross-links of your proteins. So it essentially is like a meshwork that glues everything together. And this is going to be really great for stabilizing your proteins and kind of keeping the integrity of your tissue intact. And so it's really going to be the best when you're trying to do tissue IF. But one of the downsides of using formaldehyde is that because you're cross-linking and essentially creating this meshwork, it can mask certain epitopes, and that really is a problem if that epitope that you're interested in is the one that's masked because now your antibody can no longer bind to it. The other issue that formaldehyde fixatives can have is that they can cause quite a bit of autofluorescence. So if you, for example, fix for too long, um, what can happen is that you can have these residual aldehydes that remain, and these aldehydes can then gain fluorescent properties and lead to background autofluorescence. Your other option for fixation is going to be using some sort of organic solvent. So, for example, like ethanol, methanol, or acetone. And the way that organic solvents work is that they're actually going to dehydrate the cells, and this essentially is going to dissolve any sort of lipids, and then it causes these proteins to then precipitate and denature. And so, one of the nice things about doing this type of fixation is that it doesn't mask any epitopes because you don't have to worry about any cross-linking or anything like that. But organic solvents, of course, can be quite damaging, right? Because you're essentially dissolving all the lipids and then your proteins are denaturing. So it can affect certain epitopes. So again, this is going to be an issue now if the epitope that was destroyed is your epitope of interest, and now your antibody can no longer bind to your protein. So we do recommend that anytime that you are trying out a new antibody to try different fixatives because different fixatives obviously can affect epitopes differently. So here's an example right here of just that. So our team looked at cells that were fixed with PFA versus methanol. And you can see when looking at the mitochondrial marker TOM20, PFA fixation worked much better in this situation, whereas for E-cadherin, methanol fixation worked much better. So after your fixation step, you might need to do an antigen retrieval step. And this is really going to depend on how you fix your tissue. So if you've used a formaldehyde fixative and your antigen is now masked, you need to do some sort of antigen retrieval. So the way that antigen retrieval works is that it's designed to break any sort of protein cross-links that are masking your epitopes of interest. And there are two main ways to do antigen retrieval. So these are going to be heat-induced epitope retrieval and proteolytic-induced epitope retrieval. So the heat-induced method is going to be more common. It tends to be a little bit more gentle. And so here what you're going to do is you're going to take your sample and you're going to heat it up in a microwave or a pressure cooker. And you're going to most commonly be using some sort of citrate buffer at pH 6, or you can also use a tris EDTA buffer at a slightly higher pH. So your tris EDTA buffer is going to be really beneficial if you're using antibodies against, for example, like phosphotyrosine residues. So you'll heat this up for about 20 minutes, and then that should hopefully unmask any of your epitopes. However, if you're still having difficulty unmasking your epitopes, you can go the proteolytic route. Because this tends to be a little bit harsher, it can damage certain tissues. So you just have to be really careful with the proteolytic version. So with your proteolytic version, you don't heat up your samples at all. So what you would do is you would take your sample and you would incubate it in proteinase K and trypsin for about 10 to 15 minutes. Your next step is going to be permeabilization, depending on how you fixed and where your protein of interest is localized. So this is going to depend on those two things. So, how you fix your tissue in the first place. So again, if you're using a cross-link fixative like formaldehyde, you might need to permeabilize. Also, you need to consider, where your protein is localized. So if it's an intracellular protein, you essentially need to break the membrane a little bit so that your antibodies can actually penetrate through and bind your protein of interest. So there are a few ways that you can permeabilize your samples. So you can use something like a strong non-ionic detergent like Triton. And what this will do is that this will allow you to then essentially poke some holes in your cell membranes, and then your antibody can go through and bind to your protein of interest. It's really going to be ideal if you're looking at any sort of intracellular or like or organelle specific proteins. But if you aren't looking at or organelle specific proteins, if you're just kind of looking at cytoplasmic proteins, you might be able to get away with a milder detergent. For example, you could use something like digitonin as well. I also do want to note that if you are using any sort of organic solvent as your fixative, then permeabilization is not going to be required. And this is because the process of dehydration when you're using an organic solvent, that essentially permeabilizes the cells because you are dissolving all those lipids. And so that in itself creates those pores that are going to be necessary for your antibody to penetrate through and bind to your protein of interest. The other thing that doesn't require permeabilization is if you're using formalin fixed paraffin embedded or FFPE tissues. So yes, the formalin fixation part, of course, is a formaldehyde based fixation, but the process of preparing that tissue for paraffin embedding also has a dehydration step. And so again, during that dehydration, you're creating those pores. So for organic solvents and FFPE tissue, permeabilization is not going to be required. The other thing that we recommend is that you optimize the length of your permeabilization, and this is going to depend on the thickness of your samples. So thick tissues or organoids or spheroids, these might require longer permeabilization times, but you want to ensure that you don't permeabilize for too long because you don't want to damage your tissue as well. So after permeabilization, you'll have your blocking step. So blocking is going to be crucial in order to prevent any sort of non-specific binding that can occur from your antibodies. So of course, the antibodies that you are using are hopefully going to have a really strong affinity to your protein of interest. But some antibodies can also have weak interactions with other different proteins in your sample. So you just want to ensure that you're blocking any of those potential binding sites. So generally, when you're blocking, the most common thing to use is going to be serum from the host species of your secondary antibody. So for example, if you're using a goat anti-mouse secondary antibody, you would use goat serum to block your samples. If you don't have serum available, you can also use BSA. So generally, it's recommended to use about 2 to 5% BSA, and this also works really well for blocking in immunofluorescence. It's also important, I want to note before I move on that when you are blocking, you want to make sure that the sera that you are using is not the same species of your primary antibody. So what your primary antibody was raised in. And the reason behind this is because you'll essentially lose the specificity of your secondary antibody because it will essentially bind to not only your primary antibody, but then everything else that your block has also bound to. So after your blocking step, you're ready for your primary antibody incubation. So when selecting your primary antibody, it's important that you know that not all antibodies are going to be suitable for all applications. So you might find an antibody that's going to be validated for Western blot that isn't validated for IF. So you just want to make sure that when you're selecting an antibody, you're going onto that supplier website and just ensuring that the antibody that you're using is validated for immunofluorescence. And this is going to largely depend on what type of epitope the antibody can recognize. So for example, for Western blot, these antibodies tend to recognize linear epitopes. But for IF, you need an antibody that's going to recognize conformational epitopes. So you'll find that there are some antibodies that, either work for Western blot or IF, or there's also some antibodies that can recognize both types of epitopes. But you just want to ensure that the one that you're using will work for your specific experimental condition. You also want to make sure that you optimize your antibody concentrations and incubation time. So again, going onto that supplier website is going to be really helpful. So usually most product pages will tell you kind of a recommended dilution. However, this is going to be, different from sample to sample, but it is a good starting place. So of course, the company might not necessarily have validated in the same sample type that you're using, but you at least can gauge, what type of titrations to use or where to kind of start off. The other thing you want to consider is whether you want to use a polyclonal, monoclonal, or recombinant antibody. So I'll talk a little bit more about this on the next slide because antibody selection for IF is very important. So when selecting your primary antibody clonality, there are kind of three main flavors that you can select from. So you have your polyclonal antibodies, which are going to be a mixture of antibodies that recognize multiple epitopes on the same antigen. And the way that polyclonals are produced is that an animal is going to be injected. Generally, it's a rabbit, and it's injected with a specific immunogen, and then it has an immune response, and you collect that serum. And because you're just kind of collecting that serum, it's a mixture of antibodies. And so, because it's a mixture of antibodies, that can bind to multiple epitopes. And so, it's really going to be the best option for low abundant targets because essentially multiple antibodies can bind to one protein. And it's just a way of amplifying the signal a little bit. Um, but it can also have a high lot to lot variability, right? Because anytime you run out of a lot, you need to re-immunize the animal, or if the animal dies, you need to immunize another animal. So you can see, how this can essentially generate quite a bit of variability because with the second animal, you don't know if it's going to have the same immune response as that original animal. And because of that, polyclonals aren't always the best for reproducibility. I won't say that not they're not reproducible, but they're not the most reliable. And the other thing is that they can also have quite a bit of high background since within the mixture of your antibodies, some are going to have really high affinity for your epitopes, but some might have not as high of affinity and might be binding to other things in your sample. Your second option is going to be your monoclonal antibodies. So monoclonals are specifically going to recognize one epitope on an antigen. And the way that monoclonals are made is that they're going to be produced by creating a hybridoma from the B cells of an immunized animal with a cancer cell line. And so because it recognizes one antigen with very high specificity, it does tend to produce lower background. But it's not going to be great for those low abundance proteins because you are relying on that one binding site, right? So with polyclonals, you have multiple antibodies that can bind to your protein, but with the monoclonal, you just have that one that is binding to that antigen. So you want to make sure that, when you are using a monoclonal, that your protein is expressed more widely. Another disadvantage of monoclonals is that there is an issue with mouse on mouse staining. So, for example, if you have a mouse tissue and you're using a mouse monoclonal antibody, there is the possibility that, when you add in your secondary antibody, that secondary antibody might bind to not only your primary, but also to endogenous IgGs in your sample. So, it's not ideal for mouse on mouse staining if you're doing traditional IF. Um, Another kind of antibody that you could consider is recombinant antibodies. So, your recombinant antibodies are going to recognize one epitope with a very high precision. So, recombinant antibodies are generally manufactured by cloning the heavy and light chains of these antibodies, followed by in vitro expression. And so now, because you're relying on cloning, so you have the sequence, you can essentially have an unlimited supply of this antibody. So the lot-to-lot consistency is going to be really high, which makes it, really great for long-term projects and also for reproducibility. But because recombinants are a little bit newer to the game, you don't have as great of target coverage yet. So with polyclonals, the target coverage is really great. Recombinants, the target coverage is a little bit poorer right now. However, most companies, more and more companies, are really focusing on recombinants. And I know for Proteintech, it's currently a really big initiative for us to produce these kind of renewable antibodies. So as if antibody selection wasn't already difficult, but you also have to consider what supplier you're going to choose from. And that supplier might also have several antibodies against your target of interest. So the fact is that, there's just an overwhelming number of reagents out there, and antibody selection can really take hours because you have to filter through it all. So I know Poppy touched a little bit on Able AI at the beginning, but I just want to show a specific use case where you could use ABLE in order to find an antibody for IF. So for example, especially if you're using like a popular target. So here I'm using E-cadherin as my example. So if you search E-cadherin on just the regular search bar on our website, Proteintech has over 38 products. So just the traditional search generates 38 search results. That means that you need to look through 38 products to find out if it works for IF and if it works for your specific sample type. But with Able AI, what you can do is I can just go on. Okay, I think I have to stop my laser and then click this. There it is. Okay, so I can just go on to the Proteintech website and I can start typing into Able AI and say, can you suggest an E-cadherin antibody that is validated for IF in rat tissue? And Able's going to go through his entire thought process. He's going to scan our database. And then he narrows it down to 2 products. In this situation, he recommends 2 different recombinant antibodies. And you're also going to get that validation data as well. So now you have 2 antibodies that you can choose from that work in your sample type and are also validated for immunofluorescence. So I would really highly recommend that anytime you're looking for a new IF antibody, that you consult Able on what products to use for your specific experimental needs. So another tool that I know that Poppy had mentioned in the beginning is our 3D epitope mapper. So this is also a really valuable tool that kind of streamlines your antibody selection. So with this tool, what you can do is you're able to see precise antibody binding sites on 3D protein structures. So this is going to be particularly valuable. Sorry, I keep bringing up the laser pointer. It's going to be really valuable if you need an antibody that recognizes, for example, the C terminal of a protein. So you can just move around the antibody or the protein and you can select the exact antibody that you want. Versus, for example, if you need an antibody against the N terminal of the protein, again, you can move that structure around and you can find that appropriate antibody. All right. So after your primary antibody incubation is going to be your secondary antibody incubation. So when you're selecting your secondary antibody, you want to make sure that the secondary is compatible with the host of your primary antibody and that it's also compatible with the isotype. So you want to select a secondary that is raised in a different species from your primary antibody that your primary antibody was raised in. So for example, if you're using an anti-mouse primary antibody, you want to select something like a goat anti-mouse secondary antibody. You also want to consider the isotype of the primary antibody. So for example, mice have different subclasses of IgGs, and your primary antibody might be specific for a specific IgG. So for example, like IgG1. So when you're selecting a secondary antibody, if you want to make sure that, it recognizes at least IgG1 if it doesn't already recognize all the mouse IgGs. You also want to consider your fluorophore that's on your secondary antibody. So you want to make sure first that it matches with the filters that are on your microscope, just to ensure that you can visualize your data. And then the other thing that you want to make sure is what type of dye is on the secondary. So is it a synthetic dye versus a traditional dye? So synthetic dyes, such as Alexa Fluor or our in-house Corelight dyes, these are going to be brighter and more stable. So they're going to be better for lower abundance targets, because if you need to do longer excitation times, you want to make sure you have something that's going to have more integrity. However, there are also reasons why you might want a traditional dye. So for example, you might want to use something like FitC or Texas Red. For example, if you're trying to photo bleach, you might want to use a more traditional dye for these types of experiments. You also want to consider cross-reactivity. So you want to make sure that the secondary antibody that you're using is validated for the species of the sample that you are working with, and that it also doesn't cross-react with other species. So again, going onto that manufacturer website and just double checking and seeing the kind of specificity of your secondary. An expert tip that we have is that when you are selecting a secondary, you just want to select one that is cross-absorbed, or you can go the recombinant route. So for example, at Proteintech, we have our multi-Rab recombinant secondaries, and these virtually show no cross-reactivity. And I will highlight and talk a little bit more about these in the next section. So today I've talked a lot about indirect immunofluorescence. So the idea of using a primary and then a secondary antibody. But in addition to indirect IF, there is also direct IF. So that's when your primary is conjugated to the fluorophore. So no secondary step is needed. So based off of the schematic that I'm showing you right here, I feel like the pros and cons of direct and indirect IF are pretty obvious, right? So with indirect IF, you are going to have a brighter signal because multiple secondaries can bind to your primary. So you essentially have this kind of complex of fluorophores. With indirect, you're kind of limited. With direct, you're kind of limited to whatever fluorophores are going to be conjugated to your primary antibody. However, you do kind of eliminate that need to do the secondary antibody step, so it is a quicker protocol. But I do just want to kind of go over all the pros of going direct. So another pro of going direct is that it does lead to lower background. So when you're using a secondary antibody, you do have that risk of your secondary binding to endogenous IgGs in your sample. But with a conjugated antibody, that antibody will only be binding to your protein of interest. So it eliminates that risk of off-target binding that you can see with secondaries. So same kind of thing for the second point here, where it says perfect for same species. So if you have mouse tissue and you want to use mouse primary antibody, this is going to solve that issue from your secondary antibody binding to any endogenous IgG in your sample, right? Because your conjugated primary antibody is just going to bind to your protein of interest. So you don't need to worry about kind of that mouse on mouse staining. And then again, faster and simpler protocol because you're eliminating the need to do that secondary antibody incubation step. It's also ideal for multiplexing, especially same species multiplexing. So if you want to use multiple antibodies on your sample and they're all raised in the same species, that's going to be completely fine if they're pre-conjugated, because your conjugated antibodies are only going to be binding to your protein of interest and you don't have to deal with multiple secondaries. And it does work quite nicely for high abundance proteins. So sometimes you don't want that secondary complex because it might, kind of blow up your signal and make it a little intense. So going direct does work a little bit better with some of these high abundance proteins, so you can get a little bit less of that signal intensity. All right. So for the next section, I'll talk about some reagents that we have available at Proteintech that can help obtain high quality IF data. So at Proteintech, we have quite an extensive portfolio of products that can be used for IF. So for example, we have our over 12,000 primary antibodies that have been validated for IF. This includes our polyclonals, monoclonals, and our UniRab recombinant antibodies. And just a small note about our recombinant antibodies. So our team actually uses a state-of-the-art genetic engineering technique, which we've coined as our ABCE platform. So this platform actually allows us to design antibodies that recognize specific epitopes with really high precision and excellent lot-to-lot consistency. And what makes our recombinant antibodies special is that we actually can offer high throughput production. So this platform for us is fairly quick and it's very cost effective, meaning that our recombinant antibodies are also priced quite fairly. We also have our conjugated antibodies. So we have over 3,000 conjugated antibodies for direct IF and multiplexing. We also have secondary antibodies. This includes traditional polyclonal secondaries as well as our multi-Rab recombinant secondary antibodies and nanosecondaries. And then if you are looking for a conjugated antibody, but one is not available for you commercially, we also offer our flexible antibody labeling kits, which allow you to label your own antibody in your own lab. And we also offer a couple products for IF signal amplification, including our nano boosters and our new tyramide signal amplification reagents and kits. So I'll just go into a little bit more detail on some of those products that I just highlighted. So the first is our primary antibodies that are conjugated to our coralite dyes. So our coralite dyes are bright fluorescent dyes that deliver high photostability and minimal fluorescent bleed through. This makes them really easy to combine two or more dyes in one experiment. So right now we have antibodies that are conjugated to these four dyes here. So 488, 555, 594, and 647. And our dyes are going to be equivalent in brightness and spectral profile to Alexa Fluor dyes. As I mentioned, if you are looking for a conjugated antibody, but say you're working with like a niche target and you can't find an antibody that's already conjugated, or you just can't find the color that you want, you can label your own antibody using our flexible antibody labeling kits. So what makes flexible so great is that it's actually compatible with any primary antibody. So it doesn't need to be an antibody from Proteintech. It can be an antibody from any supplier. And it works with any antibody concentration and formulation. So you don't have to worry about, whether your antibody is in glycerol or has azide. You won't have to do any sort of buffer exchange, which you sometimes need to do with traditional methods of antibody labeling. And during that process, so if you do need to do buffer exchange for traditional methods, during that process of buffer exchange, you can essentially lose a lot of quite a bit of your antibody. So these kits actually make the process of labeling much more cost effective. The other benefit of these kits is that you can label just as much as you need. So if you want to label 0.5 micrograms for an experiment, you can do that, or you can scale up if you need to. And again, with some of the traditional methods of antibody labeling, you need to essentially use the entire tube of antibodies. So you don't need all that antibody. If you don't need all that antibody labeled, it does become really inconvenient and expensive. It's also a really fast and easy kit to use. So the protocol as a whole only takes 10 minutes. And the way that it works is it uses a high affinity flex linker. So you have your primary antibody, and then you introduce this flex linker. So the flex linker is going to have the fluorophores that are covalently conjugated to it. And then within minutes, that flex linker will bind to that FC region of your primary antibody. And then what you do is you come in with a flex quencher, and that flex quencher will bind to any of the unbound flex linker, and that's it. So now you're ready and you have this conjugated antibody that you can use. So within 10 minutes, you can then input this into your IF workflow. Because you are conjugating these antibodies, they do allow for multiplexing using same species antibodies. So you can have, say, 3 or 4 rabbit antibodies that are conjugated to different colors and use them all in one sample. I also want to highlight our multi-Rab goat recombinant secondary antibodies. So our multi-Rabs are a multiclonal mixture of precisely engineered recombinant monoclonal antibodies. So basically, because they are a mixture of antibodies, they recognize multiple complementary epitopes on the same IgG. So the idea behind creating this product was to have a secondary that had the high sensitivity of polyclonals but have the superior specificity of monoclonals. We also have seen with these secondaries that they have minimal cross-reactivity with off-target species, and because they are recombinant, they also have unlimited supply and high lot-to-lot consistency. So they're going to really be ideal for those long-term projects and also the reproducibility of your data. And so then for the next group of products, I want to highlight our nanosecondary reagents. So our nanosecondary reagents are going to be nanobodies that are conjugated to fluorophores. So when you guys think of a traditional antibody, we generally think of this guy here in the orange who has, the heavy and light chains. And it's quite big, so it's about 150 kilodaltons. But some animals, like alpacas, camels, and some sharks, in addition to having this traditional antibody, they also have a heavy chain only antibody. And on that heavy chain only antibody is a binding domain, which we show here in the green color. And that's what we call our nanobody. So we can essentially extract that nanobody, and the nanobody is really small. It's only about 15 kilodaltons, so it's about 10 times smaller than your traditional antibody. And you can make that, you can take that nanobody and you can conjugate it to fluorophores. And that's what we call our nanosecondary reagents. And so these nanosecondaries are then going to recognize the Fab and FC portions of your primary antibody. So because nanosecondaries are so small, one of the benefits is that they allow for small epitope label displacement. What that means is that if you look at this figure here on the left, when you are using a traditional polyclonal secondary antibody, um, you end up with kind of this complex of secondaries, right? Because multiple secondaries can bind to your primary. So yes, this complex can amplify your signal, but at the end of the day, some of these fluorophores, like this fluorophore all the way up here, it's really far from your actual protein of interest, right? So this isn't ideal for things like super resolution microscopy or if you're trying to understand, the spatial localization of your protein. However, with nanosecondary reagents, you can see that, these fluorophores end up being much closer to your actual protein of interest. So it gives more accurate spatial data. Also, because nanosecondaries are so small, they allow for better tissue penetration, which leads to more even staining. And then we've also observed very low background with these nanosecondaries, essentially no cross-reactivity and high subclass specificity, which makes them really ideal for multiplex workflows. We've also received customer data that where they have validated our nanosecondary reagents in super resolution microscopy, for example, in Storm and STED. And then I really quickly just want to show this video from a customer. Um, So this customer used our GFP booster, which is a nanobody that recognizes GFP. So sometimes, when you're working with transgenic animal models, you might be expressing GFP, but you don't get the signal that you were expecting, and you just need a little bit of a boost. So that's what the GFP booster does. So you can see here that our customer, they use tissue clearing to clear the mouse body, and you can see that they were able to observe these neuronal projections with really fine detail. And so this is quite, I think, quite an impressive video. So while we are on the topic of signal amplification, I also want to touch on a new product that was just launched, which is our tyramide signal amplification reagents and kits. So these reagents are 100 times more sensitive than indirect staining methods because of dye accumulation and direct labeling. So the way that TSA works is that you have your primary antibody that binds to your protein of interest. You then have your secondary antibody, and this secondary is actually going to be conjugated to HRP. So you have your HRP secondary, and then you introduce your dye-conjugated tyramide. And so the interaction of your dye-conjugated tyramide with HRP causes the tyramides to oxidize into free radicals, which then allows these dyes to form covalent bonds with nearby tyrosine residues that are on your antigen of interest. So since the signal is so strong, since it's 100 times more sensitive than indirect staining, it is ideal for low abundance targets, and it's going to be highly suitable for tissue multiplexing because multiple TSA dyes can be sequentially added through cyclical antibody stripping and reprobing. So for convenience, we do offer these reagents and kits in kind of multiple flavors. So you can just get the tyramide reagents themselves, or you can purchase them as multiplex kits, which contain reagents for the entire TSA workflow. All right. So for this last section, I just want to talk a little bit about some common troubleshooting issues that people face when they're doing IF and how to kind of go about tackling these. So before you can really begin troubleshooting your experiment, you need to ensure that you have the proper controls, right? So there are four main controls that we recommend that you run when you're performing IF. So the first is going to be your secondary only control. So your secondary only control is where your sample is only exposed to your secondary antibody. And this control is going to help you ensure that the signal that you are detecting when you're using your primary is actually specific to your target. After that, you want to make sure that you also have an endogenous control, which is going to be your sample with no primary or secondary. And this is going to show your overall background, any sort of autofluorescence or artifacts that your tissue might have. You also want to make sure that you have a positive control. So this will be a tissue that you know expresses your protein. Or sometimes, if you don't have that tissue, I remember when I was in the lab, I would use a cell line that overexpressed the protein. And this is going to help you validate any sort of negative results that you get so that you know that, your result is actually negative and not, for example, caused by your antibody not working. And then you also want to have a negative control, which will be a sample that you know doesn't express your protein of interest. So ideally, this is going to be a knockdown or knockout model. And this is going to help you reveal any sort of false positives that you see in your experiment. So we'll go through three main troubleshooting issues that we commonly see. The first is, of course, going to be battling that pesky autofluorescence. So there are multiple reasons that autofluorescence can occur. So first is fixation-induced autofluorescence that can occur from formaldehyde fixation, where you have all those free aldehydes that remain that can gain those fluorescent properties and essentially cause autofluorescence. You can also have autofluorescence from endogenous molecules. So things like blood cells can interfere and cause quite a few artifacts in autofluorescence. So there's a few things that you can do in order to kind of combat autofluorescence if this is an issue. So the first tip that we have is that make sure that you fix your samples for the minimum time required. So overfixation is really what's going to cause those free aldehydes to remain, and you're going to have more of them. So you just want to find that sweet spot for fixation time, which might require, of course, some trial and error. But say you've already fixed your tissue and you're having issues with autofluorescence and you don't want to redo the entire process. So one thing that you can try to do is try to use glycine during your blocking step. So glycine is going to actually help reduce any kind of fixation-induced autofluorescence because it can react with those free aldehydes and help neutralize them. Also, we've observed that most autofluorescence occurs in the green filter. So if you're facing autofluorescence, what you can do is you can try using a red or far red filter. So fluorophores like Coralite 594 or 650 might help, and this should hopefully eliminate some of that autofluorescence that you see. If you're dealing with autofluorescence because of red blood cells, we recommend that you perfuse your tissue. So as I mentioned earlier, for some tissues like the brain, it definitely is required. So you may need to perfuse your animal first with PBS just to make sure all that blood is out, and then perfuse with your fixative, and then further immerse that tissue in your fixative. There are also certain reagents that you can try using to reduce autofluorescence. So for example, like Sudan black or sodium borohydride. So Sudan black will be optimal for reducing autofluorescence from lipids, and sodium borohydride can reduce autofluorescence that's caused by formaldehyde. And then there's also some evidence that suggests that pre-exposing your tissue with UV light can also help reduce autofluorescence in all channels. Another issue that is commonly faced is if you see weak or no signal. So there are a few potential reasons for this. So the first is that it might be that your reagents have lost their activity and if that's the case, you just need to get a new lot of your antibody and just ensure that you're storing it properly. It's also possible that your antibody conditions are not fully optimized. So if you, you know, what you just want to do is make sure that you are testing out different titrations and different incubation times and using that supplier product page to see, what dilutions they really recommend. It's also possible that your protein of interest is just not expressed. So this is where your controls are really going to come in handy. So you just want to run that positive control to make sure that your antibody is actually recognizing your protein and that your negative data is correct. Your protein of interest might have low expression. So if that's the case, you need to just amplify your signal in some way. For example, with tyramide signal amplification, a nano booster, or with a secondary that has a brighter fluorophore. It might be an issue with epitope accessibility. So if your epitope has been masked, you might need to do some sort of antigen retrieval or even try a different type of epitope retrieval method. And then the last thing that I want to address is the opposite issue, which is high background, right? So again, your primary and secondary antibody might be the culprit here. So again, just making sure that you are testing different titrations and incubation times. You might have non-specific antibody binding. If that's the case, then you just want to make sure that you prolong that blocking step, and you can also, try to mess around with the concentration of your block as well. You might also be having issues with your secondary antibody. So it might be binding to endogenous IgG. So if that's the case, you may need to perfuse your tissue or consider going direct and doing using pre-conjugated antibodies. You may need to also just do something as simple as, wash your samples a little bit more. So we didn't really talk about washing today at all, but it's definitely an important step to optimize in order to kind of minimize any sort of background. So just make sure that, your samples are thoroughly washed and try prolonging those wash steps if you still see high background. And then another thing is just to make sure that your slides are not drying out. So if your slides are drying out, generally what you'll see is you'll notice kind of like a hazy background stain. And if that's the case, you'll just want to use something like a humid box or a hydrophobic pen to just ensure that your sample is staying on your slides and it's not evaporating or falling off and drying out. So I think that is my last slide. So that is everything that I have for you guys. I know that was a lot of information that was thrown out quickly. So hopefully I can clarify or expand on anything during the Q&A. But now I'm going to just stop sharing and I'm going to pass it over to Poppy again so that she can moderate. So floor is all yours, Poppy.
Thank you so much, Afrida. That was incredibly helpful, very clear, and we've had great engagement in the chat, lots of questions. So hopefully we'll be able to come on to most of your questions. But just before that, I've got a couple more slides to share with you all. So if you have found today helpful, then please be aware that we have lots of other resources available on our website to support with both immunofluorescence and all of our other immunoassays. So you can find lots of very focused blog articles, again, on our website, which cover a whole range of different topics. So we've got there just 9 tips to optimize your IF experiments, tips for immunostaining cultured cells, a wide field versus confocal microscopy. But this is just a handful of the many we've got. So please do go and have a look at those if you're looking for something specific. We also have an entire application page on our website directed to immunofluorescence staining. We have a technical guide for download, and we have protocol videos on our YouTube channel and on our website. So again, if you've got any further questions that we don't cover today, you might find the answers in there. And I also just want to share with you all that everyone who has attended Immunoassay Week will receive a follow-up email that will have a recording of this webinar, a certificate of attendance, and your own promo code for 25% off one full-size primary antibody. So I've said this in the chat a little bit already today, but please do allow up to 1 week for this email. It will come out next week after all of the Immunoassay Week webinars have been recorded. They'll have the recordings of all the webinars. And please do check your spam folder as well, because your certificate of attendance might have ended up in there. So as I've already mentioned, if you want to learn more, check out our website as well as our blogs. We have a big video hub with lots of other focused webinars, some with external speakers as well. We've got a really great one with the Manchester Brain Bank to help with sort of the preliminary tissue sectioning step. We have our online learning portal where we have lots of free online courses where you can work at your own pace and learn techniques such as statistics. You can learn Introduction to R, and we also have a Western blotting course. And finally, as I've already said, today is just one of many webinars we have this week for Immunoassay Applications Week. So if you're not signed up to any of the others and you'd like to be, make sure that you do that this week. And if you have any other questions, you can contact Proteintech directly at our events email address there, or you can ask Afrida directly at afrida@ptglab.com. Please do follow us on our social media accounts to learn more about upcoming webinars and workshops. And with that, I'd just say a huge thank you again to Afrida. It's been super useful. As I say, I think we've had lots of questions. So I will just stop sharing my screen so you can see us both a bit better, and we'll come on to the questions. So I thought we could start off with maybe a little bit of a blocking masterclass, because we've had lots of questions about blocking. So I'll put a few of these together, but we've had questions of people asking, what do you recommend for blocking? Should it be serum, or can you block with BSA? And what kind of percentage should it be? And should it be in media or in PBS?
All right, yeah, that's a great question. So in terms of blocking, your best option is going to be serum. BSA works, it's fine, but serum is kind of going to be the gold standard. Your serum should be raised against the species that your secondary antibody was raised in. So commonly this is going to be like goat serum. You could also probably get by with, like, using FBS or something like that. In terms of concentration, this also kind of varies. I've been in labs that used between 5 to 10% serum, depending on what was passed down to them from protocol to protocol, from generation to generation. So this might be something that you need to optimize and just kind of look into the literature as well. And then I think there was one more question within that. Did I answer all of them?
When people are blocking, should the serum be in, or the BSA be in media or PBS?
Yes. So it's most common to put it in PBS.
Amazing, thank you. Yeah, I think you sort of highlighted already, but you might have to do a little bit of optimization here to figure out what works best for you, a little bit of reading the literature depending on your target protein. You did already answer another blocking question in there about whether or not the blocking serum should be raised in the same species as the secondary. And yes, yeah, you said absolutely it should be.
Yes, that's ideal.
Yeah, and then one final blocking question then is it possible to block for too long?
Yes, it is possible to block for too long, because the way that blocking works is it's almost like competition assay a little bit, I guess, right? So you're competing for those binding sites. And so if you block for too long, you might potentially block sites that your antibody would bind to. So you want to, it's important to find again that sweet spot. So going into that literature and figuring out how long you should be blocking for. But yeah, you can, I think it's hard to do, but you can absolutely block for too long.
Great, thank you. Good to know. So moving away from blocking then, we've got lots of other questions. Someone has asked, are you able to incubate overnight with a primary conjugated antibody?
Yeah, you should be able to. Again, this is going to depend based off of the antibody and maybe also the fluorophore. You might get higher background for with certain fluorophores, but your if you go onto that product page, it should tell you, how long you can incubate for. And if you're unsure, you can always reach out to the company and ask. But you should be able to do your incubation similar to how you do your primary incubation for your traditional IF protocol.
Great, thank you. So we've had a question. Do you have any recommendations for permeabilizing if you're staining for two proteins? Let's say one's cytoplasmic and nucleic and the other is a membrane receptor.
Yeah, okay, so you're talking to the right person. I had to do this for grad school. So I was working with um, a growth factor and their receptor, which it was the same thing where it was a membrane-bound receptor, but it could get cleaved, and so there was also intracellular components. And my suggestion would be first to start with a milder detergent. So start with saponin. See whether or not you can get some of that membrane staining with the cytoplasmic staining. I know you mentioned that there's also nuclear staining. So what you might ultimately have to do is first do your membrane staining and then go in with your kind of using Triton and permeabilize after the fact and do like another round of staining.
Great. Well, I'm really glad that they've managed to have the expert here to help answer their questions.
It was
Um,
very painful.
yeah, you sound like you're speaking from experience, that's for sure. So another slightly interesting question then. Somebody has asked, when you're staining on collagen gel or other hydrogels, it can be difficult to make the antibody infiltrate into the gel, and it can also be difficult to take the image under the confocal. Do you have any suggestions for this?
Oh man. I unfortunately do not have this experience. Um, so I'm not 100% sure how to answer that. I don't know, Poppy, if you have any experience in that.
I don't myself, but one thing I will say is possibly worth trying is we've had a lot of feedback from users of our nanosecondaries who say that our nanosecondaries are really, really good at infiltrating through 3D tissue cultures. So it's potentially, we can see whether or not we've had any validation of getting nanosecondaries to get through gels a bit better. And maybe that could be a potential way of at least avoiding a secondary antibody. Or you could also, I guess, try a primary conjugated if you're currently trying to use indirect staining, because again, you're just reducing the amount of antibody that has to pass through. But if you want to drop us an email with that, then for sure we can see whether or not we've got anything that's been validated in collagen gels and get back.
Yeah, no, that's a great point about the nanosecondaries.
Yeah, I mean they're just so small that they do penetrate a bit better. So people have had luck with that, and also because they can bind multiple primaries, then you can get quite a significant signal enhancement, which can also help as well if you're struggling to get signal through. Um, so I'm wondering if you've got any troubleshooting tips for loss of DAPI nuclear staining, if they're still seeing strong staining of other targets.
So in this situation, they've lost the nuclear staining.
Yeah.
So yeah, you can always go back and you can try staining again. You could also try to kind of increase the amount of time that you're, exposing your light on your microscope. So just that exposure time can be increased. But I think you can go back if you haven't cover slipped already and just do some incubated again in your DAPI.
Great, thank you. I think we've probably got time for maybe just one or two more questions, looking at the time. One that's come up here is someone has asked whether or not you should see any interference of your mounting media during confocal analysis, and if so, if you've got any tips to avoid that interference.
Yeah, so mounting media, we didn't really talk about it a lot today, um, but your mounting media is another kind of key aspect that needs to be optimized. Um, so in terms of what you want to kind of consider is, you obviously have your hard set mounting media and then your anti-fade mounting media, right? So your hard set is going to be better for your long-term storage. Your anti-fade is going to be better for like photo bleaching, but not all mounting medias are going to be compatible with all fluorophores. So depending on the fluorophores that you're using and kind of the microscope that you had, there might be certain mounting medias that might work better. So my recommendation would be to kind of first, if you're using like a core, ask the core what mounting media they recommend. Other than that, you can obviously, go into the literature and see if there's some more information. But did that answer that question?
Yeah, definitely. I mean, I didn't know the answer, so I feel like I've learned anyway, so thank you. And we'll end just on one more, which is oh, I've just lost it. Here we go. Do you have any strategies to help increase signal during multiplexing experiments?
Yes. Okay. So in terms of just overall signal, do you think is what they're
Yeah,
asking?
I think they didn't specify otherwise.
Okay. Yeah. So it depends. So if you're working with a kind of a large multiplex panel, you obviously don't want to do all the primary, secondary iterations. I understand that. Obviously, primary, secondary is going to be the best in terms of getting that signal intensity. So there are other ways. So for example, I talked a little bit about our flexible kits. So we actually have we launched what is now called Flexible 2.0. So these are the kits that we have out now. So these kits have more fluorophores per flex linker than you would traditionally get with for example, like a pre-conjugated antibody. So you could try using flexible to kind of get a kind of increase that signal because these kits were optimized with immunofluorescence in mind. So that would be kind of my first suggestion. The other thing that you could do is, I know I mentioned the tyramide signal amplification kits. So these kits are also going to be a really good alternative in terms of doing multiplex IF and trying to kind of increase that signal. This is going to be a little bit more complicated than if you had like your pre-conjugated antibodies, because this is going to require you to kind of go through that the cyclical antibody stripping and reprobing. But depending on how much you're trying to boost that signal, it might be kind of the best alternative for you.
That's great. Thank you so much. There have been some really insightful questions and answers there. So with that, I will bring the webinar to an end. Again, if we didn't manage to get around to any specific questions, you can either look on our website or get in touch directly. And with that, I want to say a huge thank you to everyone for attending, and a huge thank you again to Afrida for sharing all your wonderful immunofluorescence knowledge with us. And with that, as I say, I'll bring the webinar to an end, but I hope you all have a wonderful rest of your day.